Yeah, we're now live. We're now live, and yeah, technical lore, whatever you call it. Good afternoon, everyone, or good morning if you're on the East Coast. I'm very happy today, even though I just found out, to host the honorable and exceptional Professor Poyorazi today. Jota is a Cypriot national, I should say, a Greek, who is a professor at Force in Crete, and it seems like she's been there her entire career, and in fact she has been, because she only did a very efficient postdoc with Bartlett Mell and a very quick postdoc, very efficient PhD with Bartlett Mell in Los Angeles, and then a very efficient and quick postdoc in Athens, and then she was snatched away to the institute that she's at today. I think Jota is probably one of the very few people on the planet that continues in the tradition of Roll and Rinzel. So if you have any questions about cable theory, I think Jota will be the person to ask, maybe later. But if you ever want a model that concerns dendrites or axons or anything like that, essentially Jota is the go-to place, and it's a quite a unique position to be in, I think, simply because it's on vogue to do a single compartment or all the easy stuff, machine learning and whatnot, and Jota didn't choose that route. She stayed with cables and intractable questions regarding the dynamics of synapses on cables. So I'm excited to hear about that, and I'm actually also excited to hear about how it relates to machine learning, as that's one of the things that it says in her talk. So without further ado, Efkaristopoli for coming, Jota, and the floor is yours.
Thank you very much, Tim. Thank you for the kind invitation. It's great to be here. So I'm going to tell you about dendrites, and let me get started with this. So I'm sharing my screen. Hold on. And now, okay. Can you see my presentation?
We can see you, we can see the screen, and I'm going to shut off my microphone, but if there's any questions, and by the way, feel free to ask them, I will interrupt you and make sure.
Perfect.
If we lose you, I'll call you, so keep your cell phone close.
Okay, so I just wanted to say that I don't have access to the chat, so if there are any questions, I will not see them, so please interrupt me. Hi everyone, the topic of the talk today is essentially how dendrites help solve both biological and machine learning problems, and the last part is some of our recent ongoing work that I want to share with you. So I'm going to start with the motivation, dendrites. Why do we care about dendrites, and what are they in fact? The name comes from the Greek word dendron, which means tree, and you can see from this beautiful picture by Camillo Golgi back in 1973 that they actually look like trees. At the time, dendrites were thought to serve as protoplasmic prolongations, namely to provide nutrients to the cell bodies of neurons. It was not until the late 1888, when Ramon y Cajal introduced the neuron doctrine, that we actually appreciated dendrites as communication units. What Ramon y Cajal said was that essentially neurons are the basic computing units of the brain, and dendrites serve as their entry point, whereas axons serve as their outputs that communicate the information to following neurons. So why should we care about these structures? Well, perhaps the most important reason is that the properties of dendrites are modified, are altered in many brain diseases, especially those that are involved in memory loss. And therefore, we think that if we understand how dendrites operate, maybe we help point into new treatments. The other reason, I just mentioned it already, is that they are the main communication site of neurons. Over 19% of the synapses are formed within the dendrites, especially of excitatory pyramidal neurons. Their morphology and biophysics greatly influence the output of neurons. And in addition to that, the dendrites are often active. They contain a variety of ionic mechanisms that influence the response properties and can support local computations. What do we mean by local computations? Let me give you one nice example from the lab of Jackie Chiller back in 2003. So what we see here, I hope you can see my cursor. I think there's also a pointer somehow. Okay, pen, laser pointer. Okay. So what you can see here is layer 2-3, I believe, pyramidal neuron from the neocortex, and there are two electrodes very close to one of the dendrites that deliver synaptic external stimulation of these two locations, and then there's another electrode at the cell body recording the response. And what they found back then is that if you stimulate each one of these two electrodes separately, then what you get are small depolarizing responses that are seen here at the cell body. And it's only when you pair the stimulation of both electrodes together that you get this very strong, large response, which is essentially characterized by a dendritic spike. And this response is much higher than what you get if you just linearly sum the responses of the two electrodes individually. So this operation essentially says that this dendrite is boosting the inputs when the two arrive simultaneously, generating a very large response. And this can only happen when the two inputs are separated within just a few milliseconds in time. Beyond 5 milliseconds, you already lost this strong dendritic response. So essentially, the take-home message from this experiment is that dendrites can serve as coincidence detectors, precisely because they only boost coincident signals. Now, what is the mechanism for this coincidence detection? Essentially, an array of ionic channels, in this case, primarily sodium conductances that are responsible for these sharp dendritic spikes. So this is just one example of a local computation that you can appreciate allows the cell to perform an interesting function, that of detecting coincident inputs. Now, there are many different types of these dendritic spikes. For example, we have the fast-rising action potential-like ones that are underlined by sodium conductances, the calcium spikes, which are wider in their duration, slower, and they are generated in specific locations, primarily in the apical trunk of cortical neurons, and NMDA spikes, which are found typically within the thin dendrites, and they have much lower kinetics and this typical plateau-like shape. So you can imagine that if we have all this kind of variability in terms of dendritic responses, the computations that can be implemented with these dendritic spikes are likely to be equally multiple and important. Now, the question of whether dendritic spikes are relevant for behavior is one that is very important and has only relatively recently been answered. What I show here is a study done in the lab of Matthew Larcombe, a dear collaborator, where they essentially tested whether dendrites are involved in sensory perception. So we have a mouse here. This is the whisker of the mouse, and they placed the pole close to the whisker. The distance was varied so that the animal had a hard time detecting its presence, and it had to report that it detected the pole in order to get a water reward. And the hypothesis here was that the more difficult the detection task, the more likely that dendrites are involved. So they looked, while the animal was performing the task, they looked in the apical dendrites of layer 5 neocortical neurons in the barrel cortex. When the animal was successfully reporting the presence of the pole versus missing to detect it. And what they found was that during the heat trials, during the successful reporting, there was a strong response in the apical tough dendrites of these neurons. So this already suggests that the dendritic activity may be critical for behavior. But to demonstrate this conclusively, they had to perform some causal manipulations, which is what was done. They delivered inhibition in the distal dendrites of these cells in a localized manner and look at the probability detection. This is a control, this is with inhibition, and you can see that the probability of detection drops substantially and it comes back with recovery. This was all done in vivo. Another manipulation was to inhibit the distal dendrites by exciting local interneurons, somatostatin cells in this case. Again, you can see a substantial drop in the detection probability, suggesting that these distal dendrites are important for the behavioral task. They've also done the opposite, where they optogenetically stimulated the distal tract with specific, a very cool technology with microprisms, which allows you to deliver optical stimulation only within the distal band, the distal dendrites. And in this particular experiment, you also see an increase in the detection probability. So activating the distal trunk increases the detection probability. Inhibiting the distal trunk reduces the detection probability. Taking these results together, the study concluded that the dendritic spikes in the distal tough dendrites of layer 5 pyramidal neurons are causally linked to sensory perception. And as I said, this is one of the few studies that demonstrated how important the dendritic spikes are for behavior. Now, how can we systematically probe the significance of dendrites? We know that they're important now, at least we believe so. And the question is, what is the best way to do this? Already I presented some of the experimental approaches, which are very challenging, as you can imagine. So in my lab, we chose an easier way. We want to study them using computational methods primarily, although we're now starting experimental approaches as well. So today I'm going to give you a few examples of the modeling work in the lab. And the tool that Tim already mentioned is our favorite one. It's called compartmental modeling. In this compartmental modeling approach, you have a detailed morphology of a neuron, like the one that you see here, a reconstructed morphology. And you can describe this morphology using compartments, in this case, cylindrical compartments, so that we can have a 3D structure that resembles the original neuron. And then once we have this reduction into cylinders, into a cable essentially, we can use electrical circuit theory to simulate the activity, the voltage activity and the current activity within each one of these compartments. And thankfully, thanks to Oral, we have a very nice tool set that we can use to simulate conductances like sodium and potassium and Lie conductances, the membrane itself through a capacitant, as well as the different forces that operate within and outside the cellular fluid. So we have these compartmental models. And why is it important to actually go all this way to build cylindrical approximations of real neurons? That's maybe one question in your minds. So one of the reasons is because we want to have a tool that allows us to explain experimental results at the very low level of ionic conductances and synaptic mechanisms. And compartmental modeling is essentially the ideal tool for that. We also want to have a tool that can predict possible functions of the system that we're modeling, especially in cases where experiments are challenging, like, for example, reaching into the thin dendrites of brain structures, especially if these are really deep, like hippocampus, for example. We also want to use these models to guide further experimentation. We know that experiments are time-consuming, costly, and very laborious. So if we have a model that can predict where to look, this will be a major advantage for experimental work as well. And importantly, we believe that these models, if they help us understand how the system operates, they can also give some ideas to inspire developments in other fields, and in particular, machine learning and artificial intelligence. And there are many tools that allow us to do this kind of modeling out there. The most popular one, perhaps, is Neuron, and you used it extensively in the lab, but there's also Brian, which allows us to build more simplified network models, and a lot of others that you can find if you decide to go down this road. Now, I want to give you a few of the examples, that the type of models that we have developed in the lab, and we have both models for pyramidal neurons and interneurons. For example, we have a detailed C1 pyramidal neuron of the hippocampus, a layer 5 PFC pyramidal neuron, a visual neuron layer 2-3 from the V1 cortex of the mouse. And recently, we've also developed interneuron models, specifically fast spiking basket cells in the hippocampus and the PFC. These are some of the types of models we have in the lab at the very detailed morphological level. Then we also have approaches. Okay, so how to do? I don't know how to start the simulation now. I have to go back. Yeah, okay. So you can also connect the neurons together into small microcircuits like the one you see here. And this is in the neuron simulation environment. And for those that haven't ever used it, it's a very cool environment because it lets you see what happens in the simulated cells as a function of time, zoom in into various compartments, and have a very nice hand control of what you're actually simulating. But these models, the detailed ones, are computationally quite expensive. And if you want to simulate a large-scale network, it is often advantageous to reduce. So to go into models like this, where we don't have the elaborate morphology, and precisely because of the savings in computational power that come from this reduction, we can incorporate many different types of cells, interneurons, and simulate large-scale models. We also have this kind of models in the lab, which are essentially like the ones I presented before, the simplified ones, but where we can also add various types of plasticity rules. And what you see here as blobs are essentially synapses, and when their size changes is when the model is learning something, and this particular synapse is getting strengthened. It undergoes LTP. When it gets smaller, it undergoes LTD. So this is, let's say, the toolkit of the models that we have in the lab. And now the question is, how useful can these models be? What can they tell us about the dendritic function? And I want to give you a few examples with respect to the dendritic computations. So I mentioned already that dendrites are active. They contain a variety of voltage-gated conductances that can influence the way signals are integrated. What do we mean by changes in integration? Well, you can think of the dendritic compartment, for example, the green one or the blue one that you see here, as an input-output unit. What comes in is the strength or the synaptic input, let's say. What comes out is the response of that unit, the depolarization. And there are many ways this transfer function can be implemented. The most obvious one is the linear. As I increase the number of synapses that I stimulate, the response increases in a linear manner. This is the dotted line here. But there are other ways to integrate signals. One, for example, could be a sublinear integration, which you see here in blue. So as I increase the number of synapses stimulated, the response at some point bends below the linearity and it reaches a saturation plateau. This is sublinear integration. Or you can have supralinear integration, where you start out linearly and there is a threshold, and once this threshold is passed, there is some strong depolarizing response, in this case a dendritic spike, for example, which then reaches a maximum and you again have a saturation plateau. This could be a sigmoidal or a supralinear integration, if you like. And all of these types of nonlinearities have been reported in different cell types. And it is important to understand what it is that dendrites do, because if we want to describe the neuron as a computing unit, this type of transfer function essentially tells us how we should describe dendrites. Now, the dominant theory for many years was the point neuron dogma, according to which synapses get into the dendrite, but nothing happens to them in terms of processing. They are propagated to the cell body, just weighted by their strength. And therefore, this system sums essentially its inputs in a linearly weighted manner and then decides at the cell body whether to fire an action potential or not. In this point neuron dogma, we essentially assume that the dendrite is just it's like a linear function. Whatever comes in, the same thing comes out. Okay, and this is still very prominent in many and very useful in many of the simplified kind of large-scale network models. But we are here because we care about dendrites, so we want to figure out whether this is a good approximation or not, and for which cell types it might be a good approximation. So the first question we wanted to ask with models was, how do the dendrites of CA1 pyramidal neurons integrate their inputs? And this is really old work, which was done when I was a postdoc, and I'm just mentioning it because it is the basis for more recent studies. So I developed this detailed CA1 network model of a pyramidal neuron, incorporated all the types of ionic conductances and synaptic conductances that were known to be present in these cells, and then asked the simple question: is it true that inputs within dendrites are summed linearly? How can I test this question? If I put the same two, or let's say stimuli, synaptic stimuli A and B, within different locations of this model neuron, if their summation is linear, the response should be identical. It shouldn't matter where they are located. But instead, what we found was that if you place the synaptic input within a single branch, the response of the cell body was sigmoidal, and very different from when we separated the inputs across different dendrites when the response was purely linear. So these simulations essentially predicted that the individual dendrites are performing this kind of sigmoidal nonlinearities. At the time, it was a prediction, not shown experimentally, but soon afterwards, there was experimental work in the lab of Jackie Chiller and Jeff McGee showing that this was indeed the case. So the dendrites in here was the basal dendrites of neocortical neurons. They have the sigmoidal integration curve. And similarly, the oblique dendrites of CA1 pyramidal neurons also have the sigmoidal integration, which we now take for granted. We now know that the dendrites of pyramidal neurons are nonlinear integrators. Okay, so based on this work, a very talented PhD student in the lab, Alexandra Dzilivaki, asked, how do the dendrites of interneurons integrate their inputs? Interneurons are still considered by us as well as simple integrating devices. We treat them as point neurons on a regular basis. So even the fact that Alexandra wanted to ask this question was kind of surprising to me. But she thought that maybe interneurons are also doing something special. So she used the same approach. She used detailed compartmental modeling of reproduced morphologies of interneurons, fast spiking basket cells in the hippocampus and the prefrontal cortex, a total of 8 different morphologies. And she asked the same question. I should mention that all of these models are first validated against a large number of experimental data to make sure they are realistic before we start answering questions. I'm not showing any of the validation data here, but you can find them in the papers. So for now, you just have to trust me that these are good models. So Alexandra developed these 8 models, and she asked how their dendrites integrate synaptic inputs. And what she found was really surprising. She found that there are 2 types of dendrites in these interneurons, the green ones, which are capable of generating these dendritic spikes and therefore integrate their incoming signals in these sigmoidal ways, very similar to what the dendrites of pyramidal neurons do. But there are also these sublinear dendrites, which are not capable of generating dendritic spikes, and their response is constantly sublinear, so below the linearity. And you can find both of these types of dendrites in all of the 8 morphologies that she simulated. We couldn't find an obvious, let's say, distance-dependent association between the mode of integration and the dendrite that expressed it. So it's not, very simple to interpret what it is that make a particular dendrite be supralinear or nonlinear. In our paper, we nailed it down to a combination of morphological factors and ionic conductances like the A-type channel. But these are too much detail. The point I wanted to make here is that the dendrites of interneurons, these fast spiking basket cells, may also be doing some really interesting computations because of these nonlinearities. And we're very happy to find out a few months later that this double mode of the dendritic integration has also been found experimentally in not exactly the same cell type, but of the same family, PB cells. And in this case, there are 2 types of integration like ours, one supralinear in the red and one close to linear in blue. And in this case, there was also a clear difference in the localization of these branches. So the supralinear ones are found in the stratum oriens, the linear ones are found in the stratum radiatum. So why is it important to have these dendritic nonlinearities? As I said, our aim here was to come up with a good abstraction of an individual neuron. So going back to my own work about pyramidal neurons, given that we found that dendrites have these sigmoidal integration functions, we thought that we can then describe a detailed neuron as a 2-layer artificial neural network. So assign a nonlinearity similar to the one at the cell body in each of the individual dendrites. And we tested this hypothesis by asking, does this mathematical model predict the output, the detailed firing responses of our compartmental model? And the response was, yes, to a large degree. So with a correlation coefficient of 0.93, this is a good approximation of a pyramidal neuron. Now, what was interesting to ask was, is this the case for interneurons? And can we generally think of individual neurons as multi-stage artificial neural networks? So Alexandra asked the same question by putting the two types of nonlinearities that she found in the dendrites into this two-layer neural network, and she fitted the responses of the detailed neuron with this two-layer abstraction compared to a linear one, compared to the point neuron. And what you can see here is how good is that fit. And the artificial neural network, the two-layer ANN, is doing a much better job in capturing the responses of the detailed neurons compared to the linear. This is not to say that the linear is terrible. You can see some cases where the point neuron hypothesis is also probably good enough. But in all of them, the multi-layer is a better approximation. So essentially, this story tells us that it's probably, if we want to understand how neurons compute, it's better to consider what their dendrites are doing, because these dendrites are adding another level of computation. Why is it important to add another level of computation? Well, we've seen this with an older study where we measured the storage capacity of neurons that have linear versus nonlinear dendrites. And back then in 2001, essentially we measured this as the number of bits that the theoretical neuron can store, or you can think of it as the number of patterns that it can discriminate. And we find that in the simplified neuron models, you can have a boost in capacity in the order of approximately 50 times more in terms of storage capacity. So it is important to understand if neurons have active dendrites, nonlinear dendrites, because you then realize that they're capable of storing and processing much more information than they would if they were just linear devices. Of course, the most interesting question is, what is the functional role of dendrites at the circuit level? And for a computation that we would understand that is done by a neuronal circuit. So to answer this question, George Castellakis, who's a very talented postdoc in the lab, he developed this large-scale network model. It's not really large, it's just 500 neurons. So it's a small, let's say, circuit network model. And he wanted to study how the addition of nonlinear dendrites can help this network learn a particular memory. So a memory in this case is an associative memory. So it consists of two parts, let's say the CS and the US, if you like, the two stimuli. If you present them together to the network, then because of the underlying plasticity rules, that are many in this case, and I'm not going into details because the work is published. So because of the various plasticity rules, there are changes in the connectivity and the synaptic strengths within the network. And after learning, there is a subpopulation, a group of engram neurons, which respond strongly to the presentation of just the CS, right? So this is how we define learning. The memory consists of two parts. After training, presentation of just one part is sufficient to induce the same response. So this engram neurons is just a subpopulation of the entire network. And we want to study their properties with the presence of nonlinear dendrites and in the absence of nonlinear dendrites. Now, nonlinear dendrites can exist in pyramidal neurons, and this is the default since we've known that for a long time, as well as in our interneurons. And this is what we explored in this study. So we incorporated interneurons with nonlinear dendrites versus interneurons with linear dendrites. And we looked at the properties of these engram cells. The first thing we looked at was the size of the engram. So what percentage of neurons in this circuit is needed to learn one associative memory? You can see here that with linear dendrites, this percentage is over 40%. With nonlinear dendrites, is near 30%. And these are two specific synaptic distributions. It doesn't matter. Both of these are nonlinear. We also look at the firing activity of these neurons. And we can see that with linear dendrites, they fire at much higher rates compared to nonlinear dendrites. And finally, we look at the sparsity of the response. So how different is the activity of these neurons compared to the activity of the rest of the network? We want this to be very high because this is a good way to discriminate between a pattern learned and any random network activity. And you can see that with linear dendrites it's pretty good, but with nonlinear dendrites is significantly better sparsity. All of these metrics essentially represent ways to learn a single memory, the same kind of information with fewer resources, with fewer neurons, with neurons that fire at lower rates, and they induce a higher activity sparsity. So the take-home message from this part of the work was that it is important for a circuit to have active dendrites, in this case in its interneurons, because these active dendrites allow the circuit to learn the same amount of information using fewer resources, so making important savings. And we know that the brain is under strong evolutionary pressure, both with respect to its size, but also the energy required for it to run, right, and to do what we do on a daily basis. So any savings in terms of resources are especially important for our brains. Probably not for machines, but definitely for biological brains. So what else have we learned from this kind of models? I want to present another study that was done in the visual cortex, and where we wanted to understand how dendrites may contribute to the orientation preference of V1 neurons. This is a work that was done in collaboration with Stelios Mirnagis. Oh, I should say that if you have any questions and you want to interrupt me, I'm happy to be interrupted and take questions before the end of the talk. Up to you, okay?
Jutta, we do have two questions that I might briefly ask you. One was a very basic question, whether the nonlinear integration or the integration of the EPSP always goes up. Does integration always increase the EPSP on the dendrite, or do you see sometimes
yeah, of course. If you have sublinear integration, then you know there is, well, negative. You mean becoming smaller because the linear saturates, right?
Yeah.
Yes, of course. And in the study by human neurons, you will see how they get smaller. So that's one example. It's coming up later.
Okay,
yeah.
We'll wait for that, I suppose. And then the other one is, why exactly does more dendrites per neuron lead to saving resources?
Why exactly more? It's not more dendrites. It's non-linearity, right?
Yeah.
Why? Yeah.
I think there may have been. Maybe you want to go back to that slide with the three panels. Quickly clarify.
So, for example, if you look at the firing rate, right, one easy way, the most intuitive way to understand it is if you have, let's say, dendrites that suppress signals like this one, nonlinear, it will be more difficult for inputs to induce a spiking response. Therefore, this way, we can reduce the firing rate of the neuron. That's a relatively straightforward answer with respect to the mean firing rate. They are making it more difficult for the neuron to fire, for example. That's one explanation. I mean, there are others as well, but should I?
I think let's see if the question comes back, and then maybe we can go back again.
Okay, good.
Later.
Because I have a lot more to tell you.
Okay.
So let's go into, thank you guys for this question, into the V1 neurons. So this is a study that was done in collaboration with Stelios Mirnagis and Ji-Young Park, a talented PhD student in his lab. And essentially what we wanted to find out is what is the role of the apical versus the basal dendritic trees in the orientation preference of layer 2-3 V1 neurons. We know that these neurons fire at a particular orientation of visual stimuli, right? We've known that for years. And the example here, the experimental setup, was to put a mouse under a 2-photon microscope. The mouse was under anesthesia. And these graded light bars were presented to the mouse. And they looked with 2-photon imaging and measured the activity of individual sparsely labeled layer 2-3 neurons. First, when the neuron was intact, and then after ablating, completely removing with laser, the apical dendrite. So the first really interesting and puzzling finding was that you can cut completely off the apical dendrite from these neurons and nothing changes to their orientation preference. It is an example shown here. You can see this neuron likes these two orientation of stimuli. You take off the apical dendrite after a few days for stabilization. The preference remains the same, and you can see it here in a zoom function. So what they found is that there is no change in the preference of the neuron after ablating the apical dendrite, which was kind of puzzling. We couldn't understand this, although I should mention that the experiment was done under anesthesia, and therefore inputs to the apical dendrite are not that many. So we should take this into account. So they looked at the fine properties of the orientation preference, the tuning, and the OSI. And as you can see here, before and after, there is no significant change in all of the 18 ablated neurons and 17 control neurons. So the question was, what does the apical dendrite do, or how should the apical dendrite be tuned in order for these results to be to be valid? They also looked at cutting off basal dendrites. After the ablation of the apical dendrite, they removed first one and then two apical dendrites from the same neurons and look at the orientation responses. And what you can see when you start cutting off basal dendrites is now that things change. So the change in preference in the control is shown in black, and as you cut the dendrites is the red and then the peak. And you can see now that as you start removing basal dendrites, there is change in the orientation preference of the mouse. And this is also seen in the tuning width and the OSI. So cutting off the apical, nothing happens. Cutting off the basal, gradually changes start to occur. And the question is, what should be the input and the properties of these two trees for these results to be explained? So how to answer this question, Nasi Paputsiri, who is a staff member in the lab, she developed, extremely smart, for those of you who know her, she developed a detailed compartmental model of the layer 2,3V1 neuron. This is exactly the same, one of the morphologies that were used in the experimental data. And she validated this model based on experimental findings, one of which, for example, is that 60% of the synaptic inputs are found in the basal tree and only 40% are found in the apical tree of these neurons. And we also know the tuning curve of these inputs. We know that the synaptic inputs are tuned and they are drawn from distributions with a standard deviation sigma of 30 degrees based on experimental data. So she generated this model, which you can see here shows preference for the horizontal orientation. It has the maximum of firing rate here. And it has a similar tuning curve as the experimental data. So the first question Nancy wanted to ask is, why does the apical dendrite seem to matter very little to the tuning of the cell? So to answer that first question, she generated multiple experiments where she cut the apical tree. The cut apical tree is in blue. The blue is the control, while changing the tuning between the apical and the basal tree. So the first example is when they both have exactly the same tuning, so no difference in their tuning. And then she started increasing this difference to 20, 40, and 60 degrees. And she showed essentially that in this validated model, you only have tiny difference. You don't get any major differences in tuning, even if you expand this the orientation between these two trees to large differences. You have to go very large to start seeing an effect in the tuning of the basal tree. And that's because the model primarily follows the orientation that is dictated by the basal tree. It's basal tree dominant, essentially. That was the take-home message from these simulations. But now, how can you also reproduce the finding that if you start cutting off basal dendrites, the model remains still very robust? So these are the experimental data. This is the control. This is what happens when you cut the apical dendrite, very little change. Cutting one basal dendrite, you start to see things changing. Cutting two basal dendrites, then there is a significant change compared to the control. This is a change in the preferred orientation of the neuron, right? So these are the experimental data, and we wanted to come up with a model that explains all this. And we explored different combinations. So we looked at the two trees not varying in their preferences at all, and then varying at different degrees of tuning. And when we explored the situations where we cut the apical dendrite and the two basal dendrites individually. And we found that one way to explain this experimental data is that essentially you had to have very small differences between these basal dendrites. They had to be within the regime of 20 to 30 degrees. Larger differences would generate distributions that do not correspond to the experimental findings. So essentially the take-home message is that you have to have basal dendrites that are differently tuned, but that those differences are not very large to see this, to replicate the experimental findings. And now I want to get into the human neuron study, which is one of the questions was asked with respect to responses in the dendrites. This is a work that was done in collaboration with Matthew Larcombe and Albert Guidon. Albert was a postdoc in the lab of Matthew, and now he's setting up his own lab back in Israel, and he's an extremely smart guy. And what Albert wanted to figure out is how do human neurons and their dendrites integrate synaptic inputs? So to answer these questions, he used human slices from patients with epilepsy or brain tumor. Obviously, they took the healthy part. And he used the electrical recordings to stimulate dendrites and look at the responses at the cell body. And what Albert found was something very interesting and very different from what we have seen so far. So when he injected somewhere halfway into the apical trunk of these cells, he got these dendritic responses, which you can see here, that are different in the following way. They have dendritic spikes, you can see them here, but these dendritic spikes, they rise up very slowly and they inactivate sharply. And more interestingly, their amplitude, as we increase the stimulus intensity, becomes smaller and smaller and smaller. So this is one example where we have a non-monotonic response function of the dendrite, which we can see in summary here. So if you have very weak stimuli below the threshold for the induction of these spikes, you have no responses. These spikes are not generated at all. If you hit the ideal amplitude, then you have the maximum response, which is shown here at the peak. And as you increase the stimulus intensity, the amplitude of these spikes becomes smaller and smaller and smaller. This, I think, from 12 dendrites, and they're all normalized because they don't have exactly the same threshold or amplitude. So what was interesting in this case, apart from the fact that this type of dendritic spikes have not been reported in other animals, at least not yet, was what kind of computations can we solve with such a non-monotonic response function? So what we came up with was the exclusive OR gate. What is the exclusive OR problem? So you can think of it as a gate in which two pathways arrive, pathway A and B. If none of these pathways is activated, then the gate remains closed. If any of these pathways is activated, then the gate opens. But if both of them are activated together, then the gate remains closed. This is a nonlinear problem that is known to require a small neural network to solve it. It's not solvable by a point neuron, for example. And we thought that it's an appropriate one precisely because of this property of the response to become smaller for large depolarizations. So to test this hypothesis, Nancy developed a detailed model of the layer 2-3 human neuron and detailed in terms of its morphology. Note that we don't know a lot about the ionic mechanisms and their distributions in these neurons, so we only use the basics. And we did not explicitly model the conductance that generated these spikes, which we actually call DCAPs, dendritic calcium action potentials. We know that they are mediated by calcium, but we don't know the channel and we don't know its properties. So we phenomenologically describe this drop in the amplitude of the response as a function of increasing current. More current, smaller response, right? So it's a phenomenological model. And then we tested our hypothesis using synaptic stimuli, which we could not do in slices. So we have two pathways, the pathway X and Y plus some background synapses, and we stimulate them in the dendrites of these neurons and ask whether we can solve the exclusive OR problem. So what we see is that if we activate the first pathway, pathway X, then we have a pretty good response in the dendrite and a similar strong response at the cell body. This is also true for pathway Y because they are very similar. But if we activate them together, then what we see is that the dendrite has a much smaller, much weaker response now because the amplitude of these DCAPs decreases substantially, since we're now in the regime of the small responses. And respectively, the cell body also has a much weaker response. So this set of simulations shows that if you have such a mechanism into the dendrites of human neurons, then you can solve this exclusive OR problem. And what is even more interesting is the role of inhibition. We can use inhibition in two ways. We can apply it locally within the dendrites that have these dendritic, these DCAPs, or we can place inhibition, far away from these dendrites. And the response is very different. If you place inhibition within the same dendrite, then precisely because inhibition reduces the amount of local current that is sensed by this neuron, it pushes the response back into the regime where we have strong dendritic spikes, and we get a large dendritic response and a strong somatic response. So in this case, inhibition acts essentially as an excitation by pushing back the dendrite into the strongly activated regime. But if you put inhibition elsewhere, far away from these dendrites, then it can again act as its normal role as a suppression of excitation. So this also shows that having such a dendritic mechanism in human neurons not only increases its own capabilities to solve a nonlinear problem like SOR because of this transfer function, but it also increases the possibility of added computations subserved by other mechanisms, in this case, inhibition. So the take-home message from this work was that a human neuron, in fact, its dendrite, can solve the SOR problem, and because of that, we made it into the year in biology as one of the most important discoveries.
It can be tempting to think of the brain as a computer.
And I don't think
digital computers process information.
I'm going to move on, because I want to tell you about the last part that we've done in the lab, and this is the machine learning part. Should I take any questions on this part of the talk?
There's no questions right now, but maybe you can tell us where we can find the video.
Yeah, it's...
I'll Google it if you just tell me what you're looking for.
Just Google it, Quanta Magazine, here in biology.
I'll find it and I'll post the link. Thank you.
Thank you.
Keep going.
Okay, so now I've shown you a number of examples where I hope I have convinced you that dendrites are important because they provide important advantages to the biological system. And the question is, can we take some of these ideas to the machine learning regime? And there is already a lot of people looking into this question in addition to us. Actually, we are just new players, let's say, in this. So Spiros Hablis, who is a very talented postdoc in the lab, and he's doing pretty much everything from biophysical modelings to machine learning, he designed this artificial neural network where we incorporated dendrites into the hidden nodes. So instead of having a fully connected classical neural network, now we have, let's say, if we isolate the hidden layer, every neuron connects to its own dendrites and nothing else. So we have a restricted connectivity, let's say, at the node of each of our neurons. This is one of the things that we incorporated in our approach, which we then test on a classification problem, and in this case, the 10-class classification on NIST. So we want to discriminate between these 10 digits. The learning rule is the classical backpropagation, but now because we have a restricted connectivity, there is only propagation of the error from the neuron to its own dendrites and not to anything in between. We're also changing the location we sample the image. There are typical ways to sample an image will be just to do it randomly. So you randomly select pixels from the image and you feed this into your network. But we know that in biological neurons, and in particularly V1 neurons, we have this the receptive fields. So locations in the visual space where a given neuron is selective to and not another one. So we use this property from biology to also restrict the way we sample the image. Each dendrite has its own receptive field. And then we want to look at performance, but before getting there, I just wanted to give you a small calculation of what we save by adding dendrites. So the difference between the two types of networks is the restricted connectivity. In a fully connected network, you will have all the combinations between the nodes of one layer and the other. Whereas and this is the number of trainable parameters that you would get if you have deep dendrites versus having a fully connected network. So, because it may be hard to read the equations, I'll give you an example down here. If you have a 2-layer dendritic neural network with 16 dendrites, 100 somata, and a receptive field of 9 pixels, this would give you 32,000 parameters, whereas a conventional structure with the same number of nodes would come up to 2.5 million trainable parameters. These are weights, right? The weights are the trainable parameters. So you can already understand that you can have major savings in terms of the numbers of parameters that are trained by adding dendritic structure into your networks. In this case, 83 times fewer parameters. Now the question is, can you get the same performance? So what we have here is the state-of-the-art classification on NIST, the 10 class problems. The state-of-the-art right now with 1.5 million parameters is almost 100%. It's considered a solved problem. Our dendritic ANN gets as close as 98.1%. So it's not the same as the state-of-the-art, but it has only 81,000 parameters compared to 1.5 million. So we think that this is really promising. The way to go here is not to show that dendrites are better, as we've shown in our biological simulations as well, but that dendrites help you solve the same problem with much fewer trainable parameters. So this is the dendritic ANN with receptive fields. This is when you randomly sample the image, so receptive fields matter. This is the fully connected network with receptive fields and with dropout. So various instantiations of a fully connected network compared to the dendritic one. Okay, this is a 10 class classification on NIST. And then what we wanted to see is whether we can also find a function to which dendrites can provide some major advantage. So the function that we tested was continual learning, because we know that in biological brains, we don't really have 60,000 examples coming one after the other from all the different classes that we need to remember. But we typically learn, let's say, one thing, then another, and then another, and we are really good at not forgetting, whereas classical networks forget easily. So we tested one such example where we present the categories one at a time. So first the network like 0, then 1, all the way to the 10, and then we have a repeat. And check the performance of the various networks on this task. So the dendritic one, these are two dendritic ones, they are doing pretty well. Again, we're not as close to the current state of the art, but there you use much more parameters. We're doing pretty well on this task. Fully connected is terrible. Adding dropout does much better. So we were wondering whether dendrites are the biological solution to forgetting. So we want to compare essentially what happens between these two extremes. And note that this is just one task, one example, so preliminary data. I'm not saying that this is going to be the case for everything we look at, but let's look at these two extremes. If you now go at the dendrites meaning the nodes of these two networks, right? In the dendritic neural network, these are dendritic nodes. In the fully connected, they are just nodes, so there are no dendrites there. And look at which of these nodes respond to more than one classes. So what you see here are the overlaps. When you see a darker value, it means that this particular dendrite responds to classes 9 and 4, for example. So it overlaps between two classes. That's what we're seeing here. This is for layer 1 and layer 2, the dendritic and the fully connected, the two extremes. And you can already see by this picture that in the fully connected network, you have a lot of overlaps. Many of the nodes learn more than one class. Like for example, this one is involved in learning digits 6, 7, 8, and 9 together with 4. So whereas in the dendritic, these overlaps are very, very small. So we're thinking that this is a good indication that if you have dendrites into your network, you're doing a better job because these dendrites are learning a different part, a different aspect of the task. And if you compare now the dendritic model with the one with dropout, whose performance is significantly better than the typical ANN, and look at the same overlaps again, you now see that you have a much lower overlap now in the fully connected network compared to the dendritic. So this is another indication that essentially any mechanism that can push the learning to different parts of the network is probably going to help with forgetting. And dendrites are likely to be one such mechanism. So what we show here is that the overlap as a function of the different types of networks that we use. This is the dendritic. This is the fully connected, the typical one. Which one? Fully connected. The last one. The last one is the fully connected with nothing. And the fully connected with some dropout or receptive fields is doing better. So we're hoping that this is going to be true for many other learning problems, and that dendrites may have a potential of helping by reducing forgetting. So the comparison between the two networks is that having dendrites results in a much smaller network, much fewer parameters than a fully connected network, while achieving equal or even better performance than a fully connected network. By having these receptive fields in the dendrites, we help improve the performance a lot, and especially in continual learning. On continual learning in particular, this one task, right? Our model is better than any instantiation of the fully connected network. And we think that this is because dendrites are essentially pushing learning to different parts of the network. Of course, there is a significant disadvantage here as well, that it takes much longer, not much longer, but it takes longer to run this local backprop algorithm, 10 seconds per epoch compared to 30 compared to 10, so 3 times slower. So with that, I come to the end of my talk. I hope I have convinced you that dendrites of both excitatory and inhibitory neurons are important. They contribute to numerous functions both locally and at the circuit level. Modeling is, I believe, a great tool for studying dendritic computations because it lets us study questions that are difficult to capture with experimental techniques and offers really good intuition into new possibilities through the predictions that in turn lead to new discoveries. And we're hoping that the dendritic models can also inspire developments in other fields like machine learning and artificial intelligence. And we suggest that this may have to do with resource savings again, but also something related to forgetting. And if you're interested about learning all of our ideas as to how the dendritic models can serve as a bridge between different experimental levels and theoretical approaches, we have a review paper in Nature Review Neuroscience that you can look up, published in 2020. I forgot to put it here. So for that, I want to thank the members of my lab that made all this work possible. I presented the work of Alexandra, Nasi, Spiros, and George in this presentation. I want to thank our funders and mostly you for your attention. Happy to take questions.
Thank you very much for that. That was lovely. We're getting quite a few questions. I invited a few people on screen. I see Everton is already here. So maybe Everton.
Hi, Jota. Nice to you. Really nice talk. Thank you very much for sharing your work. I have a question. I think someone else asked a similar question from what I saw. If there is any other problem that this new dendritic nonlinearity that you found on these human neurons could help on. And for example, you showed the associative memory case, right? That the dendrites could help having a fear of decrease fine rate and other things, if you would accommodate this new nonlinearity in that model, for example, if it would help or make it a bit worse, I don't know.
Yeah, we're doing it right now, actually. We're essentially trying to put that nonlinearity into a circuit model and figure out what does it help, right? I have to tell you, it definitely makes a difference. I'm not 100% sure exactly what that difference is. I mean, we're looking at the data and I'm like, yeah, it's non-linear, and I don't have a clear explanation or answer to tell you, yes, we know it's doing that. We're still trying to figure out what it is the major advantage that comes out from a different non-linearity. I mean, it seems to be like, but I'm not sure, that it may allow a network to tune into more than one memories at the same time. So it may have come down to capacity issues, but we haven't explicitly looked at it. What I can tell you also is that this type of transfer function, the human one, does not only solve exclusive OR, it solves all logical gates. And that we are writing up now and should be out soon. So, I mean, in that sense, if you think about it from the theoretical perspective, it should be extremely powerful.
I think Desmonda Fricker had a follow-up question. Everton, why don't you stay? I see you have another question, but maybe first a follow-up question on the XORs.
Hello.
Hi.
Very short question. Just, do you know if people have looked actually if the same XOR computation is also present in rodent dendrites?
I don't know. Nobody has reported it. I mean, in fact, Matthew went back to their own data to look whether they found it in their own experiments in mice, and just nobody paid attention to it, right? Because they might have thought there was something wrong with the neuron or the dendrite. I don't know, but if anything has looked at that, I guess it should be out soon, right? But I don't know.
Thanks.
Matthew didn't see it.
Do you think this is unique to humans? So the paper seems to, it doesn't quite say it, but it's asking the question whether this might be what makes us unique compared to other mammals. Do you think there's something to that?
That's the selling point of the paper. I don't know if it's what makes us unique. No, I don't know. I mean, I would very much like someone, if I had the ability to do it, I would do it in mice, in rats, in monkeys, to check whether we actually see these responses in other mammals. This is the important question to ask right now. Is it there and we have missed it?
Yeah. Do you know how this would be learned? What does plasticity do with these kind of XOR gates? I mean, have you tried to use the plasticity rules that you have tried out before to see if this goes? I mean, it seems like this should go away if you use a normal plasticity rule on this.
Go away in what sense? Because
like in that you unlearn the XOR gate with a normal Hebbian rule? I don't know.
But you don't learn it here. I mean, learn it in the sense that it's the transfer function that does it, and the transfer function is fixed. What you learn is essentially how to tune the input to the right window of the transfer function, right? Because you have to place the input near the peak if you want.
That's right, exactly.
And push it away.
But do you think that would be stable in the face of plasticity?
No, and I think in fact plasticity would be the best way to actually move around the inputs so that you only place the ones that you want to have an exclusive OR onto.
No, but what is the learning rate that gets you there? This is what's the learning rule that gets you there? That's the question.
That's a good point. Yeah, that's a good question. No idea. We haven't looked into that. Hold on, have we? I mean, we've done an exhaustive parameter exploration with classical ANN kind of models to see what is the parameter space within which you can solve the various logical gates with such a transfer function. It's not exactly the question you ask, but assuming you had a plasticity rule, would it be able to find these weights, right? The answer is yes. There are parameters that can move around the space and find solutions for everything. Now, but what rule would do that? I don't know. I mean, in an ANN approach, it could be anything, backprop, mean squares rule. But in a biologically,
you haven't played.
No, we haven't. We haven't played biologically. So that's
Here's another question from the audience. Why does this not work in basal dendrites?
They didn't find it in basal dendrites.
But why?
Well, okay, I'll tell you why. My guess, right? My guess is that it's one way to couple the two trees under very restricted conditions. So it's an exclusive OR mechanism. It has a very narrow space within which you would get these dendritic spikes, right? In the two extremes, you get nothing. So maybe it's one way to have a very specific coupling between the basal and the apical tree. And to do that, to allow this communication, you want something that is sitting on the apical dendrite because it's the tough inputs that don't get to the soma. And you will gate them through such a mechanism and allow coupling. I mean, that's one hypothesis.
Desmonda, I don't want to keep you on screen, but if you want to stay, of course, please stay, or if you have more questions. Thank you for asking. Everton, you had another question, I think.
Okay, I was having trouble unmuting.
No, now you're muted. Now you're unmuted again.
Now you're unmuted, yeah.
Can you hear me?
We can hear you and see you.
Okay. Cool. I have another question from the end of the talk. So you compared the structure of the dendrites with a deep network when you have random, I think random
inputs. The inputs are randomly selected.
Yeah. So would there be a way of somehow optimizing the structure of the connection somehow, like to imposing that certain numbers would be 0, and to try to optimize this to see if you find somewhere some dendritic structure? I mean, would that be possible? I don't even know if that would be possible, but
would you find some dendritic structure?
Like if you impose in your optimization algorithms that some connections have to be 0, but you don't say from the beginning which ones are.
I mean, dropout is kind of like that, no?
But dropout, is it random or do you optimize?
It's random. It's random.
Yeah, but could you somehow optimize it? This is what I was thinking.
That's a very good question. That is a very good question. So, yeah.
Because some structure could appear if you try to optimize it. I'm not sure if anyone has done this. I'm not that familiar with the literature.
Not that I know of, but that's a really good point. So you're saying that this dendritic structure was a result of some kind of a wiring optimization?
Yeah, you could try to, I mean, if you impose this optimization into the network, you could see if that's one of the solutions of the algorithm. I mean, I don't know if that's possible, but it could.
I mean, Herman Kuhn has done a lot of stuff on this kind of stuff, but on this kind of questions, but with respect to, the growth of dendrites, right? And they have shown that this optimization, there are rules that underline the way that branches grow, and they do obey some kind of minimum cost principles. Whether this also translates into performance optimization, because that's where you would base your thing, right? I don't know, but it's a really interesting question.
Yeah, thank you.
Maybe 2 more. I should say we can also ask more questions in the post or at the podium in the Zoom room. But 2 more questions from the audience. How is the neuron doing the XOR mechanistically? Also, what is more complex than XOR that we can see in single neurons? Well,
would you call the other nonlinear operations more complex? They're all nonlinear. I don't know how you define more complex. But the NAND, for example, operation, all of the logical gates, you can solve them. So, yeah, more complex. I mean, if you can do all of them and then can do combinations of them, then, you do pretty much everything, right? It's a global approximator after that. So how does it do it mechanistically? That was the question?
Yeah.
Well,
I think this is the calcium channel, right?
So you have these conductances that when they respond, they generate the dendritic spikes with increasing amplitude. And essentially the neuron has to find the parameter space, so the strength of the weights of the inputs, to position a given stimulus within the response curve of these decaps. So you need plasticity to move around your inputs in the, not to move them around, to increase and decrease their strength. And these inputs also have to be located near the generation of these dendritic spikes. That's the mechanistic way. And you have channels there and they open and that's it. I mean, it's nothing more fancy than what we already know for these neurons.
Yeah. Last question. Is the dendritic N readily implementable on perhaps FPGA hardware? Would it lead to computational cost savings?
Excellent question. We're also trying that now with some collaborators. Hopefully, I mean, that's the whole point, right? If dendrites are providing savings in networks of associative memory, and we've seen them and we've measured them, right? You would expect the same if you put it on an FPGA. The main difference, I have to say, is that spiking neural networks on FPGAs, they are designed to solve other types of problems. You know, you typically want to solve a machine learning problem of classification, let's say, which right now is better done with deep neural networks and backprop rather than with spiking neural networks in terms of the learning rules. So it's not enough just to take our spiking neural nets and put them on FPGA. We have to come up also with good learning rules that do as well as backprop, and we're far from that. You would be solving this problem.
Yeah, well, knock on wood. For now, thank you very much, Jota. Maybe I'll see you in the Zoom. I guess I'll see you for sure in the Zoom room now. I put the link into the bottom, but for everyone else, please join us if you want to say hi to Jota. If you want to, I don't know, please not Zoom bomb us, but anything else would be okay. We'll end the broadcast now, and then we'll see you on the other side.
And thanks everyone for coming, and thank you for the nice comments on the chat, the ones I've been able to see.
See ya.